Life on Earth is roughly 4 billion years old. 4 billion. That number is so large it stops meaning anything. So let me scale it down. If you compress the entire history of life on Earth into a single 24-hour day, starting at midnight, here's what you'd see. From midnight until about 5 p.m. the next afternoon, 17 hours, nothing visible happens. The ocean is full of bacteria, microscopic, single-celled, invisible to the naked eye. No plants, no animals, no movement you could see, just water, rock, and microbes. For 17 straight hours. Around 5 p.m., the first multicelled algae might appear. Still microscopic, still nothing you'd notice. At about 9 p.m., 21 hours into the day, something changes. In the sediment at the bottom of the ocean, impressions begin to appear. Large, structured, clearly not bacteria, clearly not random. Something big is down there. And then at roughly 9:10 p.m., the Cambrian explosion. Animals with eyes, teeth, shells, claws, everything we associate with complex life. All of it erupts in a few minutes. 9 o'clock to 9:10. That 10-minute window, after 21 hours of almost nothing, is what we're here to talk about today. Because in those 10 minutes, something appeared on the ocean floor that we still cannot fully explain. Organisms that don't look like animals, don't look like plants, don't look like fungi, and might represent an entirely separate experiment in complex life, one that failed completely and left almost nothing behind. To understand why the Ediacaran biota matters, you need to understand the silence that came before it. For roughly 3 billion years, from the origin of life around 3.8 billion years ago until about 800 million years ago, life on Earth was exclusively single-celled, bacteria, archaea. Eventually, the first eukaryotes, cells with a nucleus, but nothing big, nothing you could see. There's a stretch of time that geologists informally call the boring billion, from about 1.8 billion to 800 million years ago. A billion years during which almost nothing changed. The planet had life, had oceans, had continents, and the most complex thing living on it was a microscopic blob. Why? Oxygen. For most of Earth's history, the atmosphere and oceans had very little free oxygen. Bacteria don't need much. But complex, multi-celled organisms, things with muscles, things that move, things that grow, need a lot of it. Without oxygen, you can't power a large body. You can't build tissues. You're stuck being small. Around 800 to 700 million years ago, something shifted. Oxygen levels began to rise, possibly driven by the breakup of a supercontinent, which increased nutrient runoff into the oceans, which fed oxygen-producing cyanobacteria. And then, snowball Earth. Between roughly 720 and 635 million years ago, the planet froze. Not a regular ice age, a near-total glaciation. Ice sheets extended from the poles to the equator. The oceans may have been covered in ice kilometers thick. Surface temperatures dropped below -40. Life survived, barely, in pockets of open water near volcanic vents, in cracks in the ice, in the deep ocean where geothermal heat kept the water liquid. And when the ice finally melted, when the planet thawed, the oceans were flooded with nutrients. Oxygen levels spiked, and the conditions for complex life were, for the first time in 4 billion years, finally met. What happened next was one of the strangest chapters in the history of this planet. In 1957, a 15-year-old girl named Tina Negus was hiking through Charnwood Forest in Leicestershire, England. She was a fossil enthusiast, the kind of teenager who spent weekends splitting rocks and cataloging what she found. On this day, she found something unusual in a rock face, an impression, clearly organic, clearly structured, in rock that was older than the Cambrian, pre-cambrian rock. She told her geography teacher. Her teacher told her that was impossible. At the time, it was scientific dogma, near universal consensus, that complex life began in the Cambrian period, roughly 540 million years ago. Before that, bacteria, microbes, nothing visible. The rocks were supposed to be empty. Tina's finding was dismissed. A schoolgirl couldn't overturn a century of paleontology. 1 year later, in 1958, a schoolboy named Roger Mason found the same fossil in the same rock face. This time, a local geologist took notice. The specimen was examined, dated, confirmed. It was real. They named it Charnia masoni, after the forest and the boy, not the girl. Tina Negus didn't receive credit for over 50 years, but that's a different kind of injustice. Charnia was extraordinary. A frond-like organism, about 50 centimeters tall, shaped like a fern leaf or a feather, anchored to the ocean floor by a small, hold-fast disc, branching in a fractal pattern, each branch containing smaller copies of itself. It was found in rocks dated to roughly 560 million years old, deep water sediments. This thing lived on the ocean floor, in total darkness, far below the reach of sunlight, which immediately ruled out one explanation. It wasn't a plant. Plants need sunlight. Charnia lived where there was none, but it didn't look like any animal either. No mouth, no gut, no visible organs, no evidence of movement. It just stood there, on the dark ocean floor, like a flag planted in the abyss. And Charnia was not alone. Similar discoveries had been made in the Ediacara hills of South Australia in the 1940s, hence the name Ediacaran. More followed in Namibia, Newfoundland, Russia, China, Ukraine. Hundreds of specimens, dozens of species, a whole biosphere of complex organisms living between roughly 575 and 540 million years ago. A world that nobody knew existed, hidden in the deepest layer of the fossil record, just waiting to be noticed. Let me walk you through what we found. And I want you to pay attention to a feeling that's going to build as we go. A sense that something is fundamentally off about these organisms, that they don't follow the rules that everything else on Earth follows, because they don't. Charnia. We already met this one, a frond-shaped organism anchored to the deep ocean floor. Fractal branching, meaning each branch is a miniature copy of the whole, which is a miniature copy of a larger structure. This growth pattern exists in mathematics. It exists in crystals and snowflakes. It does not commonly exist in animals. Charnia had no mouth, no gut, no circulatory system, no evidence of any internal organs whatsoever. It lived in total darkness, ruling out photosynthesis. So how did it get energy? The leading hypothesis is osmotrophy, absorbing dissolved organic nutrients directly through its surface, like a sponge, but without the internal plumbing. Just soaking up food through its skin, passively, in the dark. It grew to about 50 centimeters. Some related species, like Charniodiscus, reached nearly 2 meters. 2 meters of passive nutrient absorption on the pitch-black ocean floor. Dickinsonia. This is the one that generates the most arguments. Dickinsonia was flat, very flat, oval-shaped, with a series of rib-like segments radiating outward from a central ridge, like a leaf, or a quilted mattress, or something you'd find on the surface of another planet. The largest known specimens are about 1.4 meters across, roughly 4.5 feet. A flat, ribbed disc the size of a coffee table, sitting on the ocean floor. For decades, paleontologists fought over what it was. The list of proposed classifications is almost comedic. Dickinsonia has been called a jellyfish, a coral, a polychaete worm, a turbellarian flatworm, a mushroom, a lichen, a xenophyophore, a close relative of sea pens, and, in one memorable paper, an organism unlike anything alive. In 2018, a team from the Australian National University extracted organic molecules from exceptionally preserved Dickinsonia fossils. They found cholesteroids, cholesterol precursors. Cholesterol is a biomarker for animals. Plants and fungi produce different sterols. Bacteria produce different sterols. Dickinsonia had animal sterols. So, it's an animal. Case closed? Not quite. Even if Dickinsonia is technically an animal, meaning it belongs to kingdom Animalia, it has no features that link it to any known animal phylum. No mouth, no anus, no gut, no nervous system, no evidence of locomotion in most specimens, though some appear to have left faint movement traces. It's an animal that doesn't eat, doesn't move, doesn't think, and doesn't resemble anything that any animal has ever looked like. Being technically classified as an animal doesn't make it any less alien. Tribrachidium. And now we get to the geometry problem. Tribrachidium was small, about 5 centimeters across, and disc-shaped. Its most striking feature is its symmetry. 3 arms, arranged at 120-degree angles, curving in the same rotational direction. 3-fold symmetry. Every animal you have ever seen, every insect, every fish, every mammal, every worm, every octopus, has either bilateral symmetry, 2-sided, like you, or pentaradial symmetry, 5-fold, like a starfish. Some have radial symmetry that's divisible, 4-fold, 6-fold, 8-fold. Nobody has 3. 3-fold symmetry does not exist in any known animal phylum, not one, not living, not extinct, except here, in the Ediacaran. Tribrachidium breaks a rule that every other animal in the history of Earth follows, and we have no explanation for why. It doesn't fit into any lineage. It doesn't connect to anything that came later. It's a geometric impossibility, preserved in stone. Sprigina. This one looks almost normal, at first. A small organism, about 3 centimeters long, with a distinct head region and a segmented body trailing behind. It looks, superficially, like a primitive worm or a very early arthropod. Some paleontologists have proposed that Sprigina is a bilaterian, an animal with a clear front and back, left and right, a very early member of the group that includes everything from insects to humans. Others look at the same fossil and see something else. The head might not be a head. The segments might not be true segments. The apparent bilateral symmetry might be an artifact of how the organism was compressed in the sediment. We can't tell. The fossils aren't detailed enough, and there's nothing else like it to compare to. Kimberella. Here's the one ray of hope for people who want to connect the Ediacaran to modern life. Kimberella was oval-shaped, about 10 to 15 centimeters long, and is often found surrounded by fan-shaped scratch marks on the sediment surface, as if something was scraping the bacterial mat in a systematic pattern. Those scratch marks suggest a radula, the toothed feeding organ found in modern mollusks, snails, slugs, chitons. They all have radulae. And the marks around Kimberella look an awful lot like radula traces. If that's true, Kimberella might be a very early mollusk ancestor, or at least a close relative of the group that eventually gave rise to mollusks. This would make it one of the only Ediacaran organisms with a clear connection to modern animal life. One thread. One thin, fragile line connecting the Ediacaran world to ours. Everything else. Dead ends. Ernietta. And finally, the one that might be the strangest of all, simply because of how little sense it makes. Ernietta was bag-shaped, literally, a hollow, open-topped sac made of tubular segments, about 10 centimeters tall, sitting on the ocean floor, like a natural vase. It didn't move. It had no mouth. It had no visible feeding apparatus. It had no internal organs that we can detect. It just sat there, open to the water. How did it eat? How did it reproduce? How did it grow? We have hundreds of specimens and almost no answers. Ernietta is a question mark fossilized in stone. In 1992, a German paleontologist named Adolf Seilacher proposed an idea that was, at the time, considered almost heretical. Seilacher had spent decades studying the Ediacaran biota, more time, arguably, than anyone alive. He knew these organisms better than almost anyone, and after years of trying to fit them into existing categories, animals, plants, fungi, protists, he gave up. Not because he lacked data, because the data kept telling him the same thing. These organisms don't fit. Any of them. Seilacher proposed that the Ediacaran biota represented a completely separate experiment in complex multicellular life. Not animals, not plants, not fungi. A fourth kingdom, or at least a fourth major body plan, that arose independently, flourished for 35 million years, and then went completely extinct without leaving descendants. He called them Vendobionta, after the old name for the Ediacaran period, the Vendian. His argument was structural. Look at how these things are built, he said. Really look. Charnia grows fractally, each branch a miniature copy of the whole. No animal does this. Some plants do. Some mathematical structures do. Animals don't. Tribrachidium has threefold symmetry. No animal has threefold symmetry. Not one. Ever. Dickinsonia, Ernietta, and others share what Seilacher called a quilted or mattress construction. Their bodies appear to be made of inflated, tubular segments arranged in parallel, like the chambers of an air mattress. No modern organism is built this way. Not in any kingdom. No mouths, no guts, no nervous systems, no evidence of predation or defense. They lived by absorbing nutrients, osmotrophy, through their quilted surfaces, maximizing surface area the way a radiator maximizes heat exchange. They weren't animals trying to be weird. They were something else trying to be alive. And they did it for 35 million years. Seilacher's idea was controversial. Many paleontologists pushed back hard. "They're just weird animals," was the general response. "We don't need a new kingdom." And then the 2018 cholesterol study happened. Dickinsonia had animal sterols. Animals. So Seilacher was wrong? Maybe, maybe not. Here's the nuance that often gets lost. The 2018 study showed that Dickinsonia was probably an animal, or at least closer to animals than to plants or fungi. But Dickinsonia is one organism. The Ediacaran biota contains dozens of fundamentally different forms. Dickinsonia might be an animal. Kimberella might be a proto-mollusk. But what about Charnia? What about Tribrachidium? What about Ernietta? The cholesterol study didn't test those, and their body plans remain as inexplicable as ever. The current scientific consensus, if you can call it that, is uncomfortable in its uncertainty. The Ediacaran biota was probably a mixture. Some organisms may have been early animals, weird, basal, experimental, but animals. Some may have been something else entirely. Seilacher's Vendobionta, or something we don't even have a name for. And some may fall into a gray zone that our classification system simply wasn't built to handle. We have a box labeled animal. We have a box labeled plant. We have a box labeled fungus. The Ediacaran biota looks at those boxes and says, "No thank you." Roughly 540 million years ago, give or take a few million, the Ediacaran biota vanished. Not gradually. Not in a slow decline over tens of millions of years. The fossil record shows a relatively sharp cutoff. One layer of rock, Ediacaran organisms everywhere. The next layer, gone. And in their place, the Cambrian explosion. The sudden appearance of animals with hard shells, sharp teeth, compound eyes, jointed limbs, and, for the first time in Earth's history, the ability to chase, catch, and eat other organisms. What happened? There are three leading hypotheses, and they're not mutually exclusive. Hypothesis 1. They were eaten. The Cambrian explosion brought the first true predators, animals with mouths, with jaws, with claws. For the first time in history, one organism could actively hunt and consume another. The Ediacaran organisms had no defenses, no shells, no spines, no ability to flee. Most of them couldn't even move. They were soft, stationary, and nutritious, like a buffet laid out on the ocean floor for the first generation of animals that knew how to eat. If the Cambrian brought predators, the Ediacaran was their first meal. Hypothesis 2. Their world was destroyed from below. The Cambrian didn't just bring predators, it brought burrowers. For the first time, animals began digging into the sediment, creating tunnels, mixing layers, churning the ocean floor. This is called bioturbation. Before the Cambrian, the ocean floor was covered in thick bacterial mats, dense, stable, undisturbed. These mats were the foundation of the Ediacaran ecosystem. Charnia absorbed nutrients from them. Dickinsonia sat on them. Kimberella scraped them. When Cambrian animals started burrowing, they destroyed the mats, broke them up, mixed them with sediment, oxygenated the soil, which changed its chemistry. The Ediacaran organisms didn't need to be hunted. Their habitat was dismantled beneath them. The floor, literally, was pulled out from under their world. Hypothesis 3. The chemistry changed. Ocean oxygen levels, pH, nutrient concentrations, and temperature were all shifting during the Ediacaran-Cambrian transition. Some researchers argue that the specific chemical conditions that allowed osmotrophic organisms to thrive, high dissolved organic carbon, low oxygen, stable bacterial mats, disappeared as the ocean became more oxygenated and more turbulent. The Ediacaran organisms were adapted to a specific set of conditions. When those conditions changed, even slightly, they had no ability to adapt, no nervous system to coordinate a response, no mobility to seek new habitat, no reproductive speed to evolve quickly. They were locked in, and the lock changed. Most likely, it was all three: new predators from above, habitat destruction from below, chemical change all around, a triple extinction for organisms that had no capacity to respond to any of it. 35 million years of complex life, the first complex life this planet ever produced, erased in what may have been a few million years. Maybe less. Here's what keeps me up at night about the Ediacaran. We spend so much time talking about dinosaurs. Understandably, they're spectacular. They ruled the Earth for 165 million years. Their extinction is the most famous event in paleontological history. The Ediacaran biota lasted roughly 35 million years, a fraction of the dinosaurs' reign, and almost nobody talks about them. But here's the difference. Dinosaurs were animals, familiar animals. Big, yes. Strange, sometimes. But fundamentally, animals. Bones, muscles, eyes, teeth. We understand what they were, even if we argue about the details. The Ediacaran biota might have been something we don't understand, something that doesn't fit into our categories, something that represented an entirely different way of being a complex, multicellular, visible organism, a way that was tried, tested for 35 million years and abandoned. Not outcompeted, not evolved past. Abandoned. By a planet that decided to go in a different direction. And we only know about this because a handful of organisms in a handful of locations happened to leave impressions in the right kind of sediment at the right time. Soft bodies, no shells, no bones, nothing that normally fossilizes. If the chemistry had been slightly different, if the mud had been slightly coarser, if the burial had been slightly slower, we would have no idea that any of this ever existed. 4 billion years of life on this planet. For 3.5 billion of those years, every complex organism left zero trace. The Ediacaran biota is a window, a tiny, accidental window, into one brief chapter of that silence. How many other chapters are there? How many other experiments in complex life appeared, flourished, and vanished, leaving nothing? No fossils, no traces, no memory. We talk about the fossil record like it's a history book. It's not. It's a handful of torn pages from a library that burned down. And the Ediacaran biota is one page, maybe the only page, from a chapter we were never supposed to read. What replaced them, though, what came rushing into the vacuum they left, was something faster, hungrier, and more violent than anything the planet had ever seen. The Cambrian explosion, the moment nature invented eyes, and teeth, and armor, and war. That's next time. Until next time, stay curious, stay terrified,